So my name is Jane Raper and I'm a biomedical scientist and I study African parasites. In particular, I study one called a trypanosome, and it causes a devastating disease in animals and humans in Africa. Okay, so in the early 1900s, it was discovered that there was something in human blood and in a few primates, a factor that is secreted into our blood that would allow them to kill some African trypanosomes. Not all of them, but some. And what was really interesting about the baboon was it could kill all of them. And it circulates on what is known as the good cholesterol. And so the good cholesterol, you probably know from television, is the one that you want to circulate around in your blood and pick up all the bad cholesterol that's collected in your veins and your arteries. So besides its job doing that, it's turning out that the good cholesterol has many antimicrobial properties and can kill quite a few microbes in many different ways. And the factor that we study is a very small fraction of the good cholesterol, and that carries this wonderful gene product known as apolipoprotein L1, but don't worry, you don't have to remember that. And that component has the capacity to kill African trypanosomes, which is, of course, the focus of the disease that we study. So we decided to find that piece of DNA, clone it, and give it to animals that didn't have it, and see if we could confer that resistance to those animals. And so we initiated the project using mice first as a proof of concept before we did anything with any livestock of any kind.
So my name is Joey Verdi, and I work on African trypanosomiasis here in Jane Raper's laboratory at Hunter College. So we have a baboon gene, it's called apolipoprotein L1, that we have integrated into the mouse genome. We then infect the mice that do or do not have the baboon gene with African trypanosomes to see whether or not the baboon gene is providing a protective effect. As we see, a mouse that does not have the baboon gene is riddled with these human-infected parasites. So now let's check out the mouse that does have the baboon gene. And what do you know? All blood, no parasites. We've worked on this factor for many, many years. And so once we finally had a mouse that was given the gene in the right way and did not get any parasites, it was amazing. I mean, we immediately had a bunch of meetings with everybody that we collaborate with to tell them the news, and then the project just took off.
I remember. It was great. Phoning them up and saying, I've got the gene for you. I'll never forget it. It was great.
My name is Steve Kemp. I work at the International Livestock Research Institute in Nairobi, Kenya. So we met Dr. Jane Raper, and we realized that there was a possibility there of combining our efforts and using Jane's understanding of the human mechanisms of resistance with the demand for cattle that could survive in the face of trypanosome challenge.
So we initiated this collaboration and we wrote a grant together. We went ahead with the first part of the project, which was to see if we could actually clone a Boran bull.
A Boran is an African breed of cattle that's very productive and very useful in Africa, but it's very susceptible to trypanosomiasis, and so that's the first breed that we're going to start working with in an attempt to make it resistant to the trypanosome. But we've got all the groundwork in place. We've successfully cloned an animal and shown that he's fertile, and so we know that we have the material that we need to work with Jane to introduce resistance genes.
So we did that with the Boran, and this was the first time it's ever been done in Kenya, and definitely with this breed, and perhaps in the whole of Africa. And with the help of Bill Ritchie, who cloned Dolly the sheep from Roslin, they were able to get 3 pregnancies, and 1 animal survived and is now 3 years old. His name's Tumaini, which means hope in Swahili. and he has 5 babies at this moment in time from 5 different mums. So this gives us great confidence moving forward that working with this mode of transfer of genetic material, which is called somatic cell nuclear transfer, big word, will be successful when it's time to make the animal that we hope will be resistant to disease. So to date, we have successfully made quite a few mice that are completely resistant to disease. And so we're using the very same piece of genetic material that we gave the mice, and we're giving that to the bull. And we're growing blastocysts and embryos right now. So we've got to the point where, if all goes well, we'll start doing transfer into surrogate mums in the coming month, and hopefully, like before, we'll get some pregnancies, and some of them will go to full term, and we'll have a transgenic animal on the ground within the year. Yeah.